Hi students, today we are going to discuss about one more short notes from the topic of enzymes. So first question, short answer question from enzyme topic is classify enzymes with example. Last class we have discussed about this. Second question from enzyme topic is what do you mean by coenzymes and what is cofactors? Very important. Sometimes they are asking in MCQ also, mostly in the short answer question. Usually enzyme are either simple protein or complex protein. In this complex protein, sometimes protein part is, mostly protein part is there, sometimes non-protein part is there. This non-protein part is known as prosthetic group. This is otherwise called as coenzyme. Okay. In enzyme, all the last three letters are ends up with ASE, whereas in coenzyme there is no the triplet letter ASE in coenzyme. Okay. So we have two groups, that is classification in this coenzyme. One is B complex vitamins related, another one is non-B complex vitamins related. In B complex related vitamins, mostly water soluble vitamins. From B6, that is pyridoxine, the coenzyme is pyridoxal phosphate. The B1 name is thiamine, so the coenzyme name is thiamine pyrophosphate. And B9 folic acid, the active or coenzyme form is tetrahydrofolic acid. And B3, B2, niacin and riboflavin, the coenzyme name is NAD, NADP and FMN and FAD. So these are all the examples for the classification of B complex related vitamins, right? Another classification for this coenzyme is some coenzymes, they are not related to B complex vitamins. For example, CDP, UDP, ATP, SAM, that is cytidine diphosphate, uridine diphosphate and ATP, adenosine triphosphate. SAM means sulfur containing amino acid methionine. So sulfur S activated adenosyl methionine. Then CoQ, this is actually involved in electron transport chain for the carrier transportation, CoQ and lipoid. These 5 or 6 are the non-B complex vitamin coenzymes. Okay, very important MCQs. Second one is, what do you mean by cofactors? Some enzyme requires an additional chemical compounds for their catalysis, catalytic activity. This is known as cofactors, otherwise called as metalloenzymes. That is, enzymes which needed some cofactor for their activity. Without this cofactor, the enzyme won't active. So for example, I am going to give 4 examples. In your book, more than 10 examples are there. At your free time, you please go through all the 10. Copper, this is the cofactor for cytochrome oxidase. Ferrous, iron is cofactor for catalase and peroxidase. Zinc, which is the cofactor for carbonic anhydrase. Magnesium, cofactor for hexokinase. Like that, you have to read all the 10 metalloenzymes in your textbook. Okay. The next third question is, what do you mean by immobilized enzyme and what do you mean by ribosomes? So it's nothing but just for MCQ only I am giving. Immobilized enzyme means the examples are glucose oxidase, urease, hexokinase and amylase, which is used in the strip method. Actually in the laboratory they will find the urinary glucose, urinary urea. Like that, they will be using strip method of glucose by glucose oxidase. So in your MCQ, suppose immobilized enzyme they are asking means the answers will be glucose oxidase for glucose estimation, urease for urea estimation, hexokinase again for glucose estimation and amylase for lipid estimation. Okay. Second one is, amylase is both lipids and pancreas. Second one is non-protein enzymes, that is otherwise known as ribozymes. The examples are actually ribonucleic acids act as an enzymes, that is called ribozymes. Another name is non-protein enzymes. The examples are peptidyl transferase. Second one is RNase P, capital P. Sometimes in the enzyme topic, they are asking about the therapeutic agents and diagnostic reagents. Very, very important MCQ. Under the therapeutic agents, we are having two examples, that is kinase, streptokinase and urokinase, which is used for the lysis of blood clot, thrombolysis. Second one is asparaginase, which is used for the leukemias, cancer conditions. Suppose they are asking in the short notes also, you can write these two. This is enough for your short notes. Therapeutic agents, enzyme as therapeutic agents, streptokinase and urokinase and asparaginase. Next one is some enzymes act as are useful in the laboratory or medical college like institution as a diagnostic purpose or analytical purpose. Here the examples are GOD, POD, that is glucose oxidase and peroxidase method for the estimation of glucose. Second one is urease for urea estimation. Third one is cholesterol oxidase for cholesterol estimation, and uricase enzyme is used for the uric acid estimation. And restriction endonuclease in molecular biology is useful for the recombinant DNA technology. So these are all the examples for diagnostic reagents. Some enzymes act as diagnostic reagents. Okay, bye.